All right, starting for real. My name is Fifelski. For those of you who have not met me, you received emails from me that went to your spam folder on June 6th with your camp IDs, and then you emailed my boss, who then forwarded me the emails and created a chain reaction of annoyance for everybody involved. But today I'm going to talk to you about biotechnology as it relates largely to the topic, but mostly from an impact framing sort of way, as there's quite a bit to get into the depths and the nitty-gritty of it. So to begin with, we will talk about some definitions of biotechnology. So if anybody has any in mind of a generic form that you could discuss, I will ask for that in a second. We'll trace through the history of biotechnology, including the history of bioweapons and the evolution of different scientific techniques that have made it possible. We will then go on to talk about modern and future applications of biotechnology, including its dual-use form, which will allow it to both be used for good and evil, if so desired, or even on accident. And then I will share some thoughts that I have on the topic. So what is biotechnology? Does anybody have a simple definition of biotech that you would like to share? I know nobody in our lab is reading the Biotech app. I assume maybe like one team in some other lab is, but yes. Yeah, so I heard bits and pieces about that, but I think the important thing is like the mimicking of organic compounds. We got one back here. Still can't hear you, but I'm sure it's a really smart thought. So, so here's some definitions that came from the internet, from just random. I just typed in definition of biotechnology on Google and trying to give you some great T arguments that come along with it. And we have the simplest as just the studying of living organisms to develop healthcare products and processes. Obviously, this is very limited to the healthcare sector. Investopedia is probably a little bit more thinking about the economic consequences of biotech stocks, which if you're thinking about the top 25 biotech stocks, they are mostly in the healthcare area. The Encyclopedia Britannica has a very nice definition that says the use of biology to solve problems that will make useful products. So this kind of ignores the dual use feature of biotechnology. And then Merriam-Webster's has a kind of long definition, but it talks about the use of commercial products such as pest-resistant crops, new bacterial strains, and novel pharmaceuticals. My favorite definition comes from this Trump book, Benjamin Trump, not our former president, Donald J. Trump, and just says that biotechnology is the processes of modifying an organism or a biological system for an intended purpose. So this kind of encompasses both the good and the bad of biotech. So let's go to ancient biotechnology. It goes back, it wasn't necessarily a discovery that was meant to be, but begins with the early domestication of animals and agricultural techniques. This could involve, figuring out how to breed animals in a way that is most effective for the amount of proteins, how long they will live, et cetera, in the early domestication of animals, as well as food preservation techniques. Most importantly or novelly is cheeses and curding techniques that allowed for the life of dairy products to live for longer, be more edible for a while. And this involves the souring of milk with a rennet, which is a chromosome that works to sour them and stay with some salt for longer periods. And then there's yeast, which obviously are used to make bread. In ancient times, beer was a very important thing because it was a clean form of water. And it wasn't very alcoholic, so you could consume it pretty much all day, not worry about getting stomach viruses, et cetera, because it had been boiled and cooked through. But most importantly, of these early techniques that come out of yeast is the use of vinegars, which allows for, again, the pickling of items and longer preservation, as well as the ability to clean and kill microbes without any knowledge that that's really what was happening. It was a highly effective and important thing in the evolution of humankind that people don't really talk about much. And then there's mules, which is kind of the first big attempt at biotechnology that merges, that were a female, I think it's a female horse and a male donkey reproduce. And they take chromosome 62 and 64 create chromosome 63 with this new species of animal that was created by humans for purposes on the farm and their usage. Then we get to classical biotechnology. So this is where things kind of start to really have a scientific twist to them. We're post-Enlightenment, and we start to understand how genetic transfer works thanks to Mendel and his laws of inheritance, where he studied pea plants, which is in Latinum, whatever that word is. And so was able to kind of recognize how quickly pea plants were able to change through different flowering techniques and breeding techniques that would help increase yields, make them a little bit more resistant to certain situations, as well as we also have a series of other major scientific studies or revelations that we don't necessarily know the importance of back in the 19th century. The discovery of the nucleus. White blood cells were finally discovered through some scientific method. Bacterial compounds, the existence of chromosomes, as well as just vaccines, and the ability to start keeping people from dying from just common ailments. And then, in 1919, we have a Hungarian scientist named Karl Erecki. And he's the first one to coin the term biotechnology and begins to use it for mass use on the farm, including with these agricultural applications were to figure out how to best turn raw materials into food for humans. So how to most efficiently get crops into pigs, into you. And in a very early stage, figured out how to take a farm that could have 10,000 pigs at a time and then slaughter 100,000 of them a year through biotechnology techniques and just application of different crops as well as different treatment of pigs to make it possible. So not super relevant to you all, but this is where the term comes from. And then in 1924, Morgan discovers the gene and develops the theory of the gene based off of a usage of fruit flies. So who knows anything about fruit flies? What's really kind of novel about them? Yeah, south. Yeah, a fruit fly lives for 24 hours and is just constantly reproducing. So you can track the genetic makeup of them very quickly. In a matter of a couple weeks, you can kind of see 20 generations of fruit flies come through, which is pretty cool. Except when people, mainly white supremacists, use it as a justification for eugenics, which is what happened in the United States with the idea of genetic makeup being important. And so in 1924, the U.S. Immigration Act is passed, and in all of its wisdom, starts to ban people from countries that some Americans, at least those who passed the law, thought were just not very intelligent. So this is mostly Eastern Europe, Southern Europe. Remember, this is in the context of a lot of other immigration laws that are just limiting people from coming in. But as a justification for the exclusion of people and limiting their numbers getting into the country. A few years later, we have a major discovery with Dr. Fleming, who figured out that microorganisms can divide and rule, and in the process, develop the first antibiotic, or a bacterial killer. This is one of the most monumental moments in the 20th century in human history, because it has the capability to keep people from just dying of things like gonorrhea or other ailments that just plagued people back in the 1920s. Modern biotechnology kind of begins then right around the end of World War II. Obviously, scientific collaboration during the war is mostly predicated on fighting the Nazis, and vice versa, the Nazis fighting the good guys. So it slows down scientific development on this front quite a bit. It obviously drives things related to weaponization, mechanization, et cetera, in a different direction. 1953, the double helix is discovered, and we finally have an understanding of DNA. A series of studies in the 1960s and early 1970s created the developments for monoclonal antibodies, which are capable of fighting ailments in a pretty efficient manner. We begin to figure out how to insert DNA into hosts, and that then leads to the ability to clone and create life in a test tube. So we do have some instances of cloning that are fairly famous. This is Dolly the sheep, the first cloned animal that we know of. Here's some cloned dogs. It's becoming, I guess, a somewhat common technique now for people to want to get their dogs cloned before they pass. So instead of rescuing dogs, people are just going to the next level and cloning their poodles. And then this picture would, a little less grainy, would demonstrate how pigs can go through the cloning cycle, but not super relevant for that. Then we have the introduction of CRISPR, which changes the world and will change our lives, certainly, moving forward, because many diseases and ailments that we will face might be able to be genetically fixed through use of CRISPR, which is clustered regulatory interspaced short palindromic repeats. And then Cas9 is just the CRISPR-associated 9 RNAs. So here is a short video on how CRISPR works, because I am not a scientist, I'm a debate coach.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors, like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world. Wrap your head around the implications of CRISPR with this playlist on genetics, or check out some of the other mind-blowing science being developed around the world and in the universe right now.
Nope.
From the smallest single...
Hello, I'm Kurt Fifelski. I'm an expert in Canada. Let's talk about some postmodern stuff that I learned off the postmodern generator. And real quick, blah, 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 blah. I deleted the video, so we should be able to get right there. The cool thing about biotechnology and its capabilities is it might help us be able to kind of deal with some of the ecological problems that humans face. Earth Overshoot Day is the day every year in which we have went past the point of the amount of resources that the Earth can replenish in a year. And as you can see, over the course of the last 51 years, that's about been halved. So we're dramatically, consuming the Earth, pumping out emissions, poisoning the water, getting plastics in our genes and stuff like that. And so biotechnology, though, offers an ability to deal with that in a few ways. So for one instance, shark livers. Many of the vaccines that we take are sped up with a protein that comes out of shark livers. I think it's called sequins, and it requires killing and harvesting sharks. And this includes the COVID-19 vaccine. Many of you have shark liver in your system. Congratulations. You can now go on Shark Tank. But the important thing with biotechnology is there's firms that are working to create these proteins with naturally occurring sugars. So just like sugarcane from Brazil is capable of saving sharks' lives. An 8x10 rug worth of sugar can create enough of the sequin to save something like 100 sharks. And so pretty important to the process of keeping them alive. That's not the only way that this happens. It also works for invasive species. We can fix fish or genetically change fish in a way that will kill off invasive species. And there's thoughts about doing it even in the Great Lakes with some of the invasive mussel species that we can just program them to kill off. So if you recall that aft that nobody read from last year, this will be a decent counterplan to resolve some of those invasive species. And this should also just be kind of a recall to like grade 6 biology in how this reproduction works. We can also potentially eradicate malaria around the world, or even better yet, genetically modify mosquitoes so they go extinct. As far as scientists know, there's not really a productive thing that mosquitoes do in society or for any creature, so on Earth. So we could probably potentially eradicate mosquitoes with no harm to biodiversity. That said, if there's something beneficial that mosquitoes do that scientists haven't yet discovered, it could upend the entire ecosystem and cause some type of biospheric collapse, which would be obviously horrible. Another possibility is the eradication of Lyme disease. Lyme disease, for those of you who don't live in the Midwest, is acquired by ticks. They bite you, and it sucks. And we can potentially modify ticks to remove the Lyme disease strain out of them. And then there's other just pest management techniques that can be used in order to deal with crops. Maybe you've developed a crop that no longer, I guess something that's happening at my house right now is our lilies are being eaten by these lily beetles. If we could develop a lily beetle resistant lily, I think my wife would be very happy because we would no longer be killing these beetles. I have nothing to do with that, I don't care. But digressing, we can also then make crops that require less water, less fertilizer, less pesticides, less insecticides, and be able to feed the world in an effective manner that doesn't require, it's just not resource intensive, right? What does fertilizer take a lot of? Phosphorus and gases and oils that we just don't have to mine or extract from the earth if we can come up with strong crops that will deal with it. At the same time, this allows for an adaptation technique to deal with climate change because of just the problems that climate change poses to plants and farming, and the unpredictability of it. They can be stronger against droughts, they can be stronger against storms, probably not tornadoes, but for the most part, a much more effective way to do this. Now, there's some places in the world where this isn't, it's not a common currency in a lot of places. There were the ability to just love biotech crops, including Europe, where GMOs are just banned, genetically modified organisms. So there are some political objections and difficulties to this, but so far the science seems pretty good in favor of GMOs at their ability to feed people and get around some of the problems that we will face as humanity. Another thing that we can deal with is mice. So mice came to Australia probably as stowaways on ships from England, bringing over prisoners, et cetera, and didn't really have much of a natural predator. Spread across Australia pretty quickly, and it's not uncommon for Australia to have these things called mouse plagues or mice plagues, where the earth just shakes with mice. They destroy crops, they will kill livestock and eat them. When these happen, it causes hundreds of millions of dollars a year to agriculture, and in my opinion, it's just disgusting because mice are gross. And so there's some attempts to figure out how to deal with mice by using CRISPR or genetically modifying them in a way to make that possible. A group of scientists once tried to do this by injecting interleukin-4, which is a protein that when injected can modify and kind of weaken the immune system and kill them. Their goal was to kill off all the female mice and by shutting down their immune system, and therefore just eradicating the populations fairly quickly as it spread. So they get these mice, which are supposed to be, they've been vaccinated or have already had mousepox, which is the equivalent of smallpox for mice. They inject this interleukin-4. Within I think 9 days, 100% of these mice were dead. And in the process, they discovered that it's not even effective because the burnout rate would be too fast to kill off the mice. But a realization that interleukin-4, if inserted into the smallpox vaccine, or not vaccine, into smallpox, the virus, could potentially wipe out all of humanity. Because the way it works is it shuts down the immune system and then has the immune system start attacking itself. And they were very shocked at what they discovered in this. So that's a dual use and something that we have to be afraid of. Here's some ways that we can potentially deal with biodiversity loss through biotechnology. We've talked about most of these already. Some of these words, I don't even know what they mean, so I won't talk about them. Another possibility is the idea of de-extinction, or bringing something back from extinction. which seems kind of spooky and like Jurassic Park-ish, but it's a reality. There's been movements to bring back the woolly mammoth, largely to see the justifications are how elephants will deal with Arctic situations. The logic kind of falls apart on me because soon there will be no Arctic, and there will also not be any elephants because we like their ivory. But they're attempting to bring back the woolly mammoth through that. There's also been some biotechnology companies that are working on creating dinosaurs. So we could have those in the near future, and wouldn't that be cool? So another video here, which is going to require me to figure out how to exit out of this on this process.
Imagine if a plant could detect harmful chemical compounds, such as explosive material found in groundwater, and then imagine that that same plant could relay that information quickly and directly to a user. Well, believe it or not, a team of MIT engineers has shown this plant-to-human communication is a lot closer than one may think. To demonstrate this process, the team first embedded carbon nanotubes that can detect nitroaromatic compounds, which are often used in landmines and other explosives, into the leaves of spinach plants. These carbon nanotubes emit a fluorescent signal that can be read with an infrared camera. Some of the embedded carbon nanotubes emit a constant fluorescent signal that serves as a reference, while the others emit a fluorescent signal when an explosive compound is detected. Having both makes it easier to determine if the explosive sensor has detected anything or not. They then introduced the explosive compounds to the plant's roots. Similar to how a plant would naturally sample from the groundwater, if there are any explosive molecules, it takes about 10 minutes for the plant to draw them up into the leaves, where they first encounter the nanotubes. To read the signal, the researchers shine a laser onto the leaf, prompting the embedded nanotubes to emit near-infrared fluorescent light. This light can then be detected with a cheap infrared camera connected to a Raspberry Pi, which is a credit card-sized computer similar to the computer inside your average smartphone. Then, that tiny computer sends an email to the user, alerting them of this detection. In addition to detecting pollutants, the researchers are working to employ this technology to sense environmental conditions, such as drought, or to monitor plant health.
So that is about, there's a movement to find and discover and use bomb-sniffing spinach, which is just kind of baffling. And anybody ever reads DDEV against you, just say, what would happen to the bomb-sniffing spinach? And game over, debate is done. There's also the ability to make biofuels out of biotechnology. Obviously, biofuels are important part of the consumption of liquid gases, et cetera. So we can make algae, we can make crops that are more effective and efficient at developing that, which would require less land use and just be better at the environment. The goal, obviously, for some of these companies is to help maintain the power of oil companies, because a lot of this research is being done by those, and potentially kind of derail the electric vehicle evolution, but it's still kind of important in the near future to reduce those emissions. There's also the ability through biotechnology to use carbon capture and sequestration techniques that are way ahead of what we have now at capturing carbon and storing it into the ground. One of these devices here is supposed to be the equivalent of like 1 million trees. And the cool thing about it is it just buries it in the ground. It doesn't then die and then release some of it back up, whatever's not buried into the roots. So important possibility there for biodiversity. Obviously, biotechnology is important for medicines and pharmaceuticals. Here's a series of drugs or things related to COVID that use biotechnology to get done. Those nose swabs that many of you use to test for COVID, developed by biotechnology. The PCR test, developed by biotechnology. The vaccine, biotechnology. Pretty important stuff in order to fight the pandemic. It's also being used to develop techniques that are capable of fighting previously incurable cancers and other techniques to fight cancer that do not involve the very difficult and horrible process of chemotherapy, such as immunotherapy, and it has been very effective at extending people's lives, particularly from genetic-based cancers, because they can use the CRISPR process to eradicate those genes in somebody that causes those cancers to exist. I know people that have gone through it. It seems to be a lot better than some of the previous techniques that were used. Now that everything about biotechnology is good, and pretty much more than any other field that deals with existential risks, biotechnology is really relatable to most of us because it deals with viruses, bugs, pathogens, and our health. And it's not too long, we're not too far removed from smallpox as a thing that killed, in some communities, 90% of the population, those that lacked immunity. And so we do have to be kind of thoughtful of the dual use aspects of this. And so I think the most important quote from this is just that it can be directly misapplied to pose a significant threat in what it's been done. So all of those things that we've talked about before, potentially weaponized in a horrible manner. That thing about interleukin-4 with the mice, if weaponized by somebody in a lab, could wipe out most of humanity. And a lot of people think of this in part because of gain-of-function research. Now, gain-of-function research it kind of takes advantage of the evolution of a virus. There's two ways to go about evolving a virus. You can first freeze the virus and then just kind of track it through nature to see how it evolves. Or we can use biotechnology to speed up that virus and see what we can do in the future. What is this virus going to look like? How is it going to morph? How is it going to change? And this process of mutation is something that scares a lot of people because there's a lot of biotech labs now across the world. Here we go. Interleukin-4 as a possibility of doing that. And again, paired with smallpox, pretty problematic. Now, for those of you who don't know about smallpox, it kind of spread pretty quickly. It began on trade routes in Europe to the point that if you weren't killed by smallpox, you probably had it and had become immune to it. It then, through the use of transit in ships, moves to Africa, thanks in large part to the slave trade, where it's problematic, but ships are it kills people on the boats, but the ships are slow and it burns out before the boat ultimately gets to its final destination. Late 15th century, early 16th century, navigational techniques in maritime evolves very quickly. Boats get faster. It becomes possible then, when people are leaving Europe and heading to North America, for that smallpox to stay alive. It's kind of like if you're throwing a lit match into a pile of hay. Sometimes it burns out, and the boat just keeps rolling. But now the pile stays bigger, and it's possible to get to North America. And then, as we know, it's used both intentionally and unintentionally as a way to eradicate indigenous populations of North and South America in a highly effective manner, to which some communities lost 90% of the people. Smallpox is horrible. You develop these sores. They kind of just develop around yourself. They get inside of you. They spread into your system, and ultimately you choke to death by a horrible means. So it's something that we have to think about. The 1918 flu, which we're all aware of now, right, thanks to coronavirus and its similarities to that flu. There was a community in northern Alaska where everybody died. And they were frozen just in the tundra. And scientists in 1971 and then later in the 90s decided to extract the virus from the frozen tundra in order to study it, lighting fires, melting the ice, lighting fires. Finally, they extracted some from a lung tissue of somebody who had passed. And that posed a real risk, or at least look at bioethics question of, should we be taking the risk of releasing this strain of the 1918 flu back into the wild? Turned out not to be much of an issue, but still something that is a potential, particularly as permafrost is melting throughout the tundra. There are viruses that we don't know about and don't know how to study that will eventually be released, and if captured by people, could be very problematic in its use. Obviously, biowarfare is something that we're somewhat familiar with. This is the impact thing that we talk about quite a bit, is that we'll use bioweapons, or terrorists will get bioweapons, that they'll be used against people. And here's a history of of how bioweapons have been used, whether it's poison-tipped arrows being shot at people, whether it's in World War I, the use of poison agents and mustard gas. World War II, the Japanese took plague-ridden fleas, put them in bombs, and dropped them on China in order to help, or in order to attempt to spread the plague. Iraq and the Iraq-Iran war, they're using sarin gas, pretty horrible stuff. And a lot of this stuff has been largely dealt with by the Convention on Biological Weapons, but two countries did not get rid of their bioweapons stockpiles, the United States and the USSR. So there is a lot of smallpox, for the most part, stored in the tundra of Russia, as well as at the CDC labs in Atlanta, which has yet to be destroyed and still exists as a potential. We do know that the Russians, there's a fear that Russia will use bioweapons in Ukraine, and that they have the program. Many countries have these clandestine programs, and are something that are worrisome for everybody. Here is just an example of what will happen if some of these agents spread downwind. Obviously, the most severe is anthrax, that if there's an anthrax release, it could spread and kill 100,000 people pretty quickly. And at least it's a quick death. It only takes a few days. But these other ones are still a possibility. So we'll talk about this at the end as it relates to the topic, so I will come back to that. So then the next evolution of modern biotechnology is synthetic biotechnology, which now uses engineering techniques to develop and use biotechnology, which allows for the construction and use of them from scratch. It's the next wave of biotech. This includes CRISPR type stuff. I know that there's some T cards in the T file that debates whether or not synthetic biotechnology is biotechnology, and the answer is yes. And the fear, though, is that bioterrorism would be possible as a result of the use of biotechnology. So it only takes one person from a lab to do one thing bad to cause a problematic use of biological agents. Whether this person is just radicalized and angry, like what happens in the 2001 anthrax attack, that somebody's kind of mentally unstable, starts mailing out anthrax. They worked for a biotechnology lab when they're doing it. Somebody that's down on their luck and just takes a bribe or is blackmailed in order to give terrorists a biological agent, or somebody that just decides that they're going to do it and release these things. And we have a history of these things happening. I already mentioned anthrax in 2001. In 2011, two different universities developed a new form of super H1N1. So these are the genetic makeups of a virus, and it has the possibility of killing a lot of people if it's in the wrong hands. And so there's a debate then about whether or not this information should be published in scientific journals. The two biggest ones, Nature and Science. You probably cut cards from Nature. Science is a little bit more nerdy than what we usually use. Decided against it for a while, which is much to the dismay of the scientific community, which is opposed to the ideas of censorship. But the thought is, if we publish the techniques for how they came to H1N1, it would be possible for somebody that's radicalized to then use those techniques and replicate the process. It might seem kind of far-fetched, and to many scientists, it is far-fetched about how this would work. But in 2015, scientists at the University of Alberta went online and bought all of the stuff they needed to make horse pox from just internet websites that sell you stuff. And became very apparent of the risks that biotechnology poses for a lot of people. And this is because garage biotechnology is becoming more and more common. These are people that are either hobbyists or just scientists that are working on side gigs that have biotech labs in their homes and easily purchase things that, well, yeah, there's probably some registry that's kind of tracing it, would enable somebody to develop something like horsepox with $100,000, something that can be very easily converted into smallpox, and then weaponized against people. So I don't want to ruin Power of the Dog for you, but it was a very good movie. Some of you have probably seen it. And this is kind of the process, right? This kid gets some anthrax out of a dead animal, uses it to kill Benedict Cumberbatch. Spoiler. And is an easy process that somebody with a basic level of scientific literacy is able to do. There's some studies that say that some elementary school students would have the capability now with the equipment that's available in their schools to build a bioweapon if they so decided, which is problematic. We also just have some questions of bioethics as it relates to the dual-use feature of it. Obviously in the US, we kind of take we're somewhat leery of China in some ways. And part of it's because their Made in 2025 program has biotechnology as one of the key components in order to develop their own, to lead in pharmaceuticals and to lead into science. It's also necessary because of the aging population as a result of the one-child policy. And there's some thoughts that they will then be able to use CRISPR and its related systems to genetically modify babies in a way that is more suitable for life. In fact, this scientist here cloned twins, created twins out of a test tube and with some genetic material back in like 2016. Thought it was going to go over really well, was saving his findings for a big scientific study or a conference. Comes out with it and obviously ends up losing his job and having some problems. But some of these countries, China and Russia, don't necessarily have the same scientific scruples that we have in the US. And there's many instances of people taking bribes, et cetera, to kind of to look the other way in the biotech field. Obviously, you've probably at some point heard this. I'm not suggesting that COVID was created in a lab in Wuhan and released, so don't take that away from this. But it's possible, right? It's not out of the realm of possibility that all of this could happen, and it's somewhat likely to occur somewhere around the world. Lynn Klotz, so for record, when you're cutting biotech or bioweapons, bio-stuff impacts, look up this person, kind of a kook in my opinion, and some of the like outlandish things that he thinks. But he says that there's a 24% chance of an existential level pandemic happening because of a lab in the next 10 years. And uses some pretty detailed scientific methods to go through it. These charts on the right compare his studies to Fournier's studies, who has a little bit of a difficulty in dealing with it. But it only takes one person for it to occur. And an instance of this would be, imagine for a second that you work in a biolab, you're in a Bio4 lab, the highest security facilities in the world. And you're in your spacesuit and you're dealing with things, but there's a tiny rip in that spacesuit. And the germ that you're working with gets into the spacesuit. You don't know it, but it's in your system. You leave. You go to South Quad, you're hanging out with your friends. You're talking. Every time you exhale, water vapor comes out that includes this virus. You have no symptoms yet because it takes 24 hours, and it spreads. And then your friend goes to lab, spreads to them. You all go to Starbucks, spreads throughout there. It doesn't take very long for the transmissibility to take over. And it's much like COVID. And this has happened in a couple of instances. In 1977, the flu virus was wrongly activated with H1N1, which caused people to get H1N1 from the flu virus, which complicated a mass vaccination campaign of the Carter administration. In 1978, smallpox, remember smallpox is eradicated, but some scientists are working with it. It gets leaked through air vents, killing people in a scientific study. In the USSR, there's in 1979 an accidental spill of anthrax that kills an entire city of people. 2004, a CDC research lab in Beijing accidentally leaks the coronavirus, and it causes the SARS pandemic. 2014, some scientists mailed the strains of typhoid across the country in the U.S. in through regular mail. The package was ripped when they got the vials. Some of them were broken. and we barely missed a surge of typhoid. And just generally, there's about 100 accidental releases in the US every year. And that's just because, and that's only at places that receive publicly funded, or are publicly funded. Those that are privately funded, like corporations, don't have to report that information. So a huge potential there. China's been using biotechnology as well to monitor people, mostly the Uyghurs, and their genocide of them, for lack of a better phrase. Also using these techniques in Tibet. So states have the ability to use them for negative consequences. There's some fears of a Russia-China partnership over biotechnology that would allow for them to more efficiently develop some of these capabilities. Let's not forget that one of Russia's pretenses for the war in Ukraine is that there's biotech labs that need to be invaded and prevent the Ukrainians from having access to them. And then, so the topic. The Solvency Advocate in the starter packet is really calling for a conference of sorts to start working, having governments work together within NATO to both create a repository of information about biotechnology, to create safety standards that they can all agree to and work through, and then develop the censoring techniques to prevent biotechnology from being in wrongful hands. There's other calls for how biotechnology can be used within NATO. First is cognitive biotechnology, so like rewiring people's brains through the use of biotechnology. US and NATO could kind of cooperate through that. There's these cytoskeletons, which are like futuristic uniforms for military people that can be developed. Here's an example through use of biotechnology. It's kind of like the merger of AI and biotech in its capacity to do much harm or good. But it seems like, in my opinion, a lot of the biotechnology's inclusion in the topic is a little bit, it's not necessary for NATO to really be involved if the US and these other countries can just work together on their biotech platforms and capabilities. So I look forward to seeing, I know a couple labs are working on biotech stuff in this first wave, to seeing what they've done to kind of advance or evolve the biotech research put forward at the camp. But my early take on it is it will be around, but it's not the most consequential of affirmatives. And then the real solution to all this is going back to the Convention on Biological Weapons. It's just to reform these conventions and have a little bit more international buy-in and cooperation through scientific diplomacy to get things done. Pretty much every article I read, every academic article I read in writing this presentation basically went back to those early conventions. It was just like, we need to bolster them, we need to adopt them, we need to move them forward to make them work. All right, do we have any questions? Yes, you're gonna have to speak up because I can't hear from back there. Yeah, I mean, it's developed out of biotechnology techniques. And if it's not biotech, then certainly AI, right, in the way that it works. So you have a 2AC that gets to make two different we meet / counterinterpretation arguments. Yes?
What do you think the ethical implications are of telling teenagers that they're likely capable of creating bioweapons?
Well, I mean, you all would have known that by going on the internet or reading a 1AC that said it. I mean, I will say that some of this stuff seems a little bit overstated. So this Klotz person is where a lot of this kind of comes from, and his estimates, right, a little bit more of a fearmonger type person that's fearful of these things happening. But if you have $100,000 in a biotech lab, then yeah, you could probably get it done. Yeah. Yeah, you. Yeah, so bioethics are like the big thing, or one of the big questions of what to do about biotechnology, because biotechnology has the possibility to do a lot of good for the world. It also has the ability to do a lot of evil. And then there's just these questions of like, should we bring back woolly mammoths? Like, is that ethical? Should we be making babies, letting people genetically modify babies to make sure that they have the right eye color that they want? And like, it gets murky and some of that stuff. Entire fields of study on bioethics, and certainly I think a lot of critical literature on the negative against the bioethics AF this year. For anything that's like, we need to make bioethics better, to be like, no, or biotechnology better. Let's think about the Heideggerian nature of the use of this technology. Back there, then you. So the NATO key stuff is largely US key because we have the most biotech labs, and that we need to work together to create a repository of information about the military applications and terrorist applications of biotechnology. It's light. I don't think it's the strongest NATO key warrant, but it is a warrant that the negative has to think about how they want to counterplan around. You, and then back over here.
What is NATO's current overall stance on biotechnology in general?
NATO doesn't really have much of a cohesive biotechnology policy. It's more they're afraid of like the use of bioweapons, but each country has its own independent angle on biotech. So there's not something cohesively binding them. Red shirt. You're gonna have to speak up. So the question was how can an AF deal with disparities in the application of biotechnology, whether it be income-based within the US, whether you have, what type of health insurance you have will determine whether you get immunotherapy or chemotherapy, as well as other nations that lack access to it. I think the AF's key warrant there would be that we need to, if we speed up development, like an accelerationist type approach, if we just speed it up, we can get it to more people. But certainly, it goes back to that bioethics question and is somewhat problematic. George? Can you repeat that one more time? So the U.S. any lab that the U.S. publicly funds, which is quite a few of them, it has some oversight over, and that if there's an accident or something like that, they have to report it in order to keep getting that funding and prevent from being shut down. So there is some use there, but private corporations are largely shielded from that, and I imagine many of the military applications fall in both of those categories. Like if Raytheon is working on some bioweapon or bio capabilities, there's not a lot of oversight. So an AF might say that oversight is lacking now. Working with NATO will create a new framework where that oversight would exist. Otherwise, counterplan, just do the oversight. All right, last question. I think it's one of those like, could they? Yes. Will they? I mean, they die too. So it's not as, it's a massive, it's the same like nuclear terror type question of like, do you attempt to do it? I mean, I would, there are examples of scientists working with Al-Qaeda and bin Laden to develop bioweapons. There's a bioterror attack in Japan where the subway system got flooded with something real close to anthrax killing a bunch of people. So are they likely to do it on a smaller scale, a few hundred people? I would say yes. Are they going to try to do an extinction level type capacity? Probably not. And there's some complications that go along with it. All right, well, I will be up here to answer any more questions. I think you all get a 10-minute break before our next presentation. So thank you.